Nucleic acids, this is for you, this is chemistry for you grade 11 and grade 12. So today we're going to talk about the nucleic acid. Nucleic acids are always related to generic information. All the hereditary information is not just about the shape or color of the eye or whatever you have. It's about your cholesterol levels. It's about how your body is processing sugar, whether you can be diabetic one day or not. All that information, including the construction plans for building your body, is stored chemically in compounds called the nucleic acids. So, the nucleic acids, as the polysaccharides, which are the units for building sugars, and the polypeptides, which are the units for building proteins, nucleic acids are biological polymers. Nucleic acids are formed from equal numbers of 3 chemical units: a sugar, a phosphate group, and one of several nitrogenous bases. The backbone of the nucleic acid is a sugar phosphate chain with various nitrogenous bases connected to the sugar units. So we're going to start. We have 2 types of nucleic acids: the deoxyribonucleic acids and the ribonucleic acid. Of course, the most famous is the deoxyribonucleic acid, which also known as DNA. Of course, you know that the DNA is found in the nucleus and is found sometimes in the mitochondria and the chloroplasts. The RNA is found throughout the cells. The DNA acts as the biological storehouse of the biochemical storehouse of the genetic information in the cells of all living organisms. The sugar in the DNA is deoxyribose, which has a ring in which 4 of the atoms are carbons and the 5th atom is oxygen. Here is it. Okay, 4 of the atoms are carbon and the 5th is oxygen. The phosphate group comes from the phosphoric acid, HO3PO. 2 of the OH groups from the phosphoric acid condense with the OH groups on 2 different sugar molecules, linking all the 3 together. The nitrogenous bases, they connect to the sugar units in the backbone. Here, let's look at it. These are the nitrogenous bases. Any one of the 4 bases, adenine, guanine, cytosine, and thymine, is connected along the strand at the sugar units. All the genetic information is encoded in the sequence of the 4 bases, which are also abbreviated to A, G, T, and C. Living things vary in the size and number of the DNA molecules that are present in their cells. The cells may have just 1 or many molecules of DNA. Some bacterial cells, they have a single DNA molecule that has about 8 million bases. Human cells have 46 molecules of DNA that have a total of about 6 billion bases. The DNA is a double-stranded structure, and it is a 3-dimensional structure. So it is formed as it exists as a double helix. The 2 strands are not duplicates of each other. Instead, they are complementary. This means that where an adenine is found on one strand, thymine is found on the other. And where guanine is present on one strand, it is matched with cytosine on the other. The reason for this complementary nature of the DNA is when A and T are lined up opposite each other, the 2 bases are ideally placed for forming 2 hydrogen bonds, which bond the 2 strands together. Likewise, when C and G are present in front of each other, they form 3 hydrogen bonds between themselves. No other pairing can form from the right hydrogen bonds to keep the strands together. Thus, the 3-dimensional configuration of DNA looks like a twisted ladder or spiral staircase. Now we're going to talk about the DNA replication. There is a copy of your DNA in each cell of your body because your DNA is able to replicate itself efficiently. To begin the replication, a part of the double helix unwinds. So the double helix, it's going to become a single helix, providing two strands of DNA. Each strand is going to act as a template for the making of a new strand. New nucleic acid units made by the cell are going to meet up one by one with their complementary bases on the template. The hydrogen bonds formed between the correct base pairs A to T, T to A, C to G, and G to C. As the nucleic acid units line up on the template strands, covalent bonds form between the sugars. Eventually, the original double helix is replaced by two perfect copies. As long as we're talking about DNA, we also need to talk about genes, because a gene is a segment of DNA that has the code for the amino acid sequence to build a polypeptide. The way that the gene is translated into an amino acid sequence is complicated. It uses many proteins and other nucleic acids, sometimes the ribonucleic acid or the RNA. The protein synthesis begins in the cell, making an RNA strand, which is the ribonucleic acid. The DNA double helix unwinds and RNA units match up with the DNA bases. The RNA has the base uracil instead of thymine. So whenever you see T in DNA, it is not T in RNA, it is U. So in this case, the U is going to be matching with A. So again, in DNA, the T matches with A. In RNA, the U matches with A. However, instead of using DNA unit, the cell uses RNA units which differ from the DNA by an oxygen on the sugar unit and in one of the bases. So again, whenever you see the base A and you are looking at an RNA copy, in this case you are going to match the A with U. Although in DNA copies we match the A with T. Now we're going to talk about the genetic code. There are 20 different amino acids and only 4 RNA bases. Thus, a single base cannot specify a single amino acid. In fact, a group of 3 of the bases in RNA indicates a particular amino acid. So, for each of the amino acids that are present in your body, you need 3 bases to code for them. They contain the genetic material to prepare and to cook literally this amino acid. About the gene technology. After when the scientists discovered the DNA, and then they discovered the RNA, and then they learned about how DNA replicates, they started expanding the horizons of the gene technology. They started by the mapping and the identification of the DNA using a technique which is called DNA fingerprinting. They use the DNA fingerprinting to identify where a sample of DNA comes from, as of course you have seen in many movies. In DNA fingerprinting, the scientists compare what we call autoradiographs of DNA samples. Autoradiographs are images that show the DNA pattern of nitrogenous bases. How do they identify DNA from very small samples? First of all, you have to know that no 2 persons share the same DNA except for twins, identical twins. Otherwise, we say that this person is this kid's father, or this DNA belongs to this person for according to a certain percentage of matching. Identifying DNA from small sample, how does it happen? The forensic applications of DNA fingerprinting can make use of a single hair or the smallest trace of blood. Then the scientists, they use the small samples because they can rapidly copy or amplify, increase the number of the DNA strands. By making many copies of tiny sample of DNA, they make enough DNA to see the pattern. And to do this, they use a method which is called the polymerase chain reaction, or the PCR, which replicates a short targeted sequence of double-stranded DNA, so it's going to increase its number, and then in order to do this, the components of the DNA are added to a solution that has the DNA and an enzyme and a primer. What is a primer? The primer is a short length of single-stranded DNA that has the complementary sequence of the first few bases of the target. The solution is then subjected to a number of heating and then cooling cycles. The heating is going to denature the DNA and separates the double strands. The cooling causes the primer to connect to the ends of the target. So what do? They literally plant the DNA, increase the number in order to be able to read the sample. How did cloning happen? Identical twins, they arise from the chance splitting of a group of embryonic cells early in the growth of the human baby. Remember when we talked in biology about the undifferentiated cells, which are the cells that haven't been specialized? When the cells are in a zygote, which is the step before having an embryo, these cells, they are undifferentiated. So this cell can become a bone, or it can become a heart, or can become a muscle. These cells include stem cells in animals or meristem cells in plants, which can be cultured artificially so they grow incomplete organisms. These organisms are going to be genetically identical to the organisms from which the cells were harvested. And in this case, we say that they are clones of their parents. Cloning mammal is a different task. However, it was accomplished in 1997 by the Scottish scientist, of course, Ian Wilmot. His work produced a sheep named Dolly. Dolly's genes were taken from the mammary cells of one sheep and placed in cells without a nucleus of another sheep. So they literally put the software of a cell in another cell without software, because they removed the nucleus. So this cell doesn't have a software anymore, and they planted the software in it. This is why in cloning the cells are a copy paste, because it's just your software that is transmitted. It's not like producing an organism. When you produce an organism, there is a mix-up of the genes between the parental and the motherly genes. But in case of cloning, they put the genetic information of one cell in another cell from which they remove the nucleus. Dolly's embryo was then raised in the uterus of a third sheep. The scientists have artificially cloned many other living things, not only sheep but plants such as orchids and other animals such as many kittens. And some rich people, they don't want to lose their dogs or their pets, and they require the scientists to clone their pets, and they pay thousands of dollars in order to have their pets cloned. The final thing we're going to talk about is the recombinant DNA, and the recombinant DNA has been used to cure some diseases by literally cutting and pasting the DNA. Making use of the protein that cut and reconnect the DNA molecules, the scientists have learned to insert genes from one species into the DNA of another. When this recombinant DNA is placed in a cell, the cell is able to make the protein which is coded the new code that I introduced instead of the old code, which wasn't okay or maybe was producing a disease. That's all for the nucleic acids. And by that we have finished this last chapter, and this is all what you're required to know for your test.